Okay? This is a controlled process where we still have the same amount of fuel, but we're kind of minimizing the amount that we're using at any one point in time to maximize the amount of energy we can get out of it. There's the same amount of potential energy between the gas canister and the gas in the car. But depending upon which way you choose to utilize it, you're going to get a different reaction. Okay? Does that make sense? So literally, your mitochondria are performing combustion inside of your cells at this exact moment. What's that?
Like a motor.
Exactly. Your mitochondria are acting similar to the motor. Now, I kid, I would never put any canisters of gasoline out in the parking lot for any of you. Last semester students, completely different story, but I like you guys. No, okay, just kidding. All right, so let's move on to talking about these three stages. I've kind of danced around this long enough. We're going to start looking at glycolysis, the citric acid cycle, and oxidative phosphorylation. As we talked about last time, glycolysis literally translates to the breakdown of sugar. And in this particular process, we're going to look at glucose being split apart into 2 to 3 carbon molecules called pyruvate. Once we generate that pyruvate and any byproducts produced during the generation of that pyruvate, then we're going to actually convert it to acetyl-CoA. And that acetyl-CoA is going to be used in the citric acid cycle. Now, as its name implies, it's a cycle. So it's going to grow over and over and over again, depending upon what we can put into it. We'll talk about acetyl-CoA. 2. Yeah. Pyruvate is made out of 3 carbons, and glucose has 6 carbons. So if we split glucose in half, we can make 2 pyruvates, each with 3 carbons. Finally, the process that's going to give us the most bang for our buck, and honestly, I'm being a total and gigantic nerd here, this is one of the coolest processes I've ever seen in any of my studies in biology. Okay? The way that this works is really freaking cool. So, I can't wait to get there and show you. But first, we've got to wade through glycolysis and the citric acid cycle. Okay? So, first thing you need to know. Glycolysis takes place in the cytoplasm. It is the only stage of cellular respiration that takes place in the cytoplasm, or the cytosol. The citric acid cycle and oxidative phosphorylation both take place in the mitochondria. Okay. During glycolysis, we are going to produce 2 molecules net of adenosine triphosphate through a process referred to as substrate-level phosphorylation. Yes, during glycolysis, we are going to generate a net total of 2 adenosine triphosphates through a process called substrate-level phosphorylation. Additionally, we're going to produce NADH. Where is NADH going to be used? Does anybody remember? In the electron transport chain, which we're going to use, remember, to generate the largest amount of ATP. So notice, part of the beauty of the way in which these different processes work together is that your glycolysis and your citric acid cycle are going to not only make ATP, which is good because it's energy, but they're also going to make stuff that we need to carry out oxidative phosphorylation. Okay? Now, the pyruvate generated is going to be converted, and it's going to enter the citric acid cycle. And we'll talk about that when we get there. It's going to enter the citric acid cycle, and the citric acid cycle will also be used to generate ATP. But still more importantly, what else is the citric acid cycle making? NADH and FADH2, which are both electron carriers, so where do you think they're going to be used? In the electron transport chain, which is going to help us carry out oxidative phosphorylation. The citric acid cycle, for every 1 molecule of pyruvate that enters, it can make 1 ATP. So for every 1 molecule of glucose we hydrolyze during glycolysis, we generate 2 pyruvates. So from one molecule of glucose, we make 2 pyruvates, which means I can carry out the citric acid cycle how many revolutions? 2. Which means if I'm making 1 molecule of ATP every revolution, how many molecules of ATP do I make for 2 revolutions?
2.
2 total. Okay? We're going to look more in depth at it soon. Just understand, right now, how many ATPs have I made at this point? 4. Okay? 4 out of how many I'm supposed to make? 36 to 38. So I've made a 10 of my ATPs. Yes, Alice?
Okay, so the citric acid cycle produces NAD and NADH, or is it the electron transport chain? Or is that utilized?
NADH and FADH2 are produced during both NADH is produced during glycolysis and the citric acid cycle through redox reactions. FADH2 is produced during the citric acid cycle through redox reactions. All of those electron carriers that are generated during glycolysis and the citric acid cycle will be utilized in the electron transport chain to fuel oxidative phosphorylation. Go back because I'm kind of spoiling the end of this for you before we even got there. And I'm expecting a lot of you in terms of understanding this when we really haven't even broken down what's going on in all these processes. So I promise I will hopefully be able to elucidate it for you soon enough. At the end of all of this, the NADH and the FADH2 help us fuel oxidative phosphorylation, which utilizes 2 different elements, an electron transport chain and a process called chemiosmosis to make a buttload of ATP.
So that's the rest of the ATP.
Yes, that's right. So if we made 4 here, the other 34 come from oxidative phosphorylation. And the reason why I get so excited about oxidative phosphorylation, outside of mental instabilities that my doctors told me about previously, is due to the fact that this process that generates so much ATP doesn't use a single bit of ATP. What we'll see in glycolysis is we actually have to use ATP to make ATP later on. We don't have to do that in oxidative phosphorylation, and I think that's really, really cool.
That's so wonderful.
It is.
Do we use ATP in
Citric acid cycle?
yeah.
We do not. The citric acid cycle does not utilize ATP, but notice this as well. The citric acid cycle and glycolysis only make 2 molecules of ATP for both of them. What form of phosphorylation do they use? Substrate level phosphorylation. We'll talk about that soon. Literally what it is an enzyme binds to a molecule of ADP and a molecule that has a phosphate on it, and it basically breaks the bond in the molecule that has the phosphate and transfers it over to the ADP, which requires its own amount of energy as well as a certain amount of time. Oxidative phosphorylation uses oxidative phosphorylation, which we're going to find is generated by those protons through a process called chemiosmosis. We'll talk about it. Basically what it is we're generating a chemical gradient with those protons, and that chemical gradient is going to be what drives the phosphorylation of ADP to ATP. Yes, Allison?
So I'm just trying to understand the whole pyruvate thing. So you said it's 2 3-carbon pyruvate. Now is pyruvate supposed to be like a
Sugar?
Supposed to be a sugar, right?
Yes, it's a 3-carbon sugar.
Okay, so what relates to that and the ATP?
Okay, so
The pyruvate.
let me go ahead and just give you a quick look at where we're going with all of this. Okay, here's where we're going. What we're going to do the next time we meet is we're going to take a molecule of glucose, and we're going to first start phosphorylating that glucose, but more importantly, we're going to start cleaving that glucose. And the reason why we're phosphorylating it first is those phosphate groups act as markers so that enzymes know to interact with this glucose. After we cleave it, we're then going to start to take those phosphate groups back off so we can rephosphorylate ADPs and convert them into ATPs. The pyruvate that's left over we're going to convert into something that will enter the citric acid.